All right, seventh graders, this is part 2 of your DNA lab station. So first and foremost, you guys need to open up this link right here. And it's going to take you guys into this. Before the days when we had Adobe Flash, it used to be a little bit more interactive. I'm not too stoked about the way it's set up now. But you guys can go ahead and click download, and it's going to turn it into a PowerPoint for you guys. So that will pop up right here at the bottom of your screen. You guys can click on that. And basically what you guys have is you guys get to look a little bit into how detectives and forensic scientists use DNA to figure out who maybe committed a crime if there's any DNA evidence that's left behind at a crime scene. So before we move on to this, let's just take a look at some of the questions you guys are going to answer. Okay, what were 4 pieces of evidence that gave us DNA? So there's a lot of things that we can use as evidence, but not everything can give us DNA to use as evidence. So that's the first question for you guys. Next one, what process do we use to multiply DNA found in a crime scene? So if you guys find a little piece of hair, you guys might find DNA at the follicle of that hair if the follicle is included, but it might not be enough to actually take a look and compare it to anyone else's. So we have to use this process where we take a strand of DNA and we replicate it millions and millions of times. So now we have something that's big, that we have enough of it to examine and we can take a look at. Okay, it's again hard to just look at like a couple strands of DNA and be able to interpret them, but if you have a lot, we can cut them into fragments and kind of study them that way. Okay, which suspect matched the sample from the crime scene? Again, I'll go through that with you guys when we get to that point. And what is the probability that 2 samples match? So when I say loci or locus, that means at one spot in the DNA. So what's the probability that 2 samples will match in only one spot? So if we're comparing like Johnny's DNA to Bethany's DNA, okay, what's the chance that they're going to match at one little spot in their DNA? Okay, what about 4 spots in their DNA? What's the chance that they match in 10 different spots in their DNA? So that's kind of what that question's answering. And again, you guys will get the answer out of this PowerPoint. Okay, so a crime has been committed. It's up to you to try and solve it. In this activity, you'll take a closer look at a crime scene, decide what evidence to collect, and how to get it back safely to the lab. Learn more about DNA profiling and compare different profiles to see if you can help to solve the crime. Good luck. So what you guys will see is different things like this. Again, this used to be a really cool interactive website. Now it's just a PowerPoint because Adobe Flash went away. Okay, so look closely at this crime scene. What do you think has happened here? When you guys see questions on the right side, I just want you to discuss them with your partner. So what do you guys think happened here? What could you collect as evidence? Make a list. Again, you can just kind of make a mental list of all the things you guys would want to take back to the lab. So check out different things you guys can see. This is a piece of gum, a baseball hat, coffee cup, cigarette. You guys can kind of check out the window. There's a little piece of shirt right here. So I just kind of want to point out some of these things that you guys can't see anymore. You used to be able to click on them and it would zoom in on it and you could read about it. Okay. All right. So think about the samples on your list. Which of you, so make sure you guys kind of do these little questions before you move on to the slides. Think about the samples on your list. Which of these do you think contain DNA? Okay. Again, think about that. Write down your ideas. Okay. And then how do you leave DNA behind? So this is kind of how we would leave DNA behind. So if we all leave DNA behind wherever we go, DNA is the molecule found in the nucleus of every cell in your body. Therefore, any sample containing cells will contain DNA. Think about the samples on the previous slide. Would you like to change any of your answers? Okay, so you guys are going to keep reading through this. I'm not going to go through all of this. This is kind of where you guys get to read about replicating DNA to make it big enough. So that's the answer to, I believe, question number 3. Okay, and then when you guys get down to here, what you guys have is DNA profiles. So what we do is when we've replicated and have enough of our DNA using PCR, we use a chemical that breaks our DNA up into specific segments. And then we can compare how many segments there are and how big those segments are using this process called gel electrophoresis. And what gel electrophoresis is, it gives us this kind of barcode. Okay? So this barcode is how we can kind of compare. All right, this is the sample from the crime scene. So this is obviously the criminal's DNA. You guys can see its unique barcode based on its DNA. You can look at roommate number 1. Did they commit the crime? Was it roommate number 2? Or you guys can compare suspect 1, suspect 2, suspect 3, suspect 4. So you guys can compare the 2 roommates to see if either of those could be the culprits, or we also picked up 4 suspects. So again, here's that same sample from the crime scene. which one of these looks exactly like this sample from the crime scene, if any. So that's going to be your guys' answer for question, I think, 3. Okay, let's go check. Yeah, question 3. Sorry, the last one was question 2, not question 3. Okay, and then finally you're going to read about how likely is it to have a person match in one DNA. So if you have 2 people's DNA, you and some other person clear across the world, what's the probability that you match your DNA with them in one spot? It's actually really common, so there's a 1 in 10 chance that you guys, so if you guys compare yourself to 10 random strangers, chances are you match with them in at least one spot in your DNA. So 1 out of 10, you guys will have one spot where you match. Okay? In 4 spots, if you matched with 7,000 people, one of those people would match with you in 4 different spots. Okay? 10 loci, if you guys went and found a billion people in the world, okay, you would match with one of them in 10 different spots. So that's really rare to match with someone in 10 spots. Okay, so that's your answer to your last question there. Okay? All right, so that's going to conclude your guys' part 2 crime scene DNA lab station.